Okay, I think we can get started. Thanks for coming, everyone. If you've been to some past ones, you've probably heard a talk from Kathleen Torkko before. Um, they're very good statistics talks, and I think she has another one scheduled in September as well. So there'll be a couple more.
Okay. Well, maybe after I do this one, you'll rescind that invitation. No.
I don't think so.
Okay. So shall I start?
Yeah, go ahead.
Okay. Well, today my topic is statistics and studies using cancer cell lines. And I'm specifically going to talk about the topic of generalizability, independence, and bias, or actually I change that a little bit to discuss small sample sizes. Now, my previous lectures for this seminar series have been very applied and practical. This one I'm going to call more of an essay, and it's based on my experiences over the past 20 plus years that I have had the privilege of working with basic scientists and analyzing their data from their bench studies. I am a population scientist. I do have bench experience, worked in labs for some time, but not with cell lines. But I do work with people who work with cell lines. So to start this journey through statistics and the intersection with the data from cancer cell lines, I just want to start with this beautiful figure. I'm always amazed when I open up papers and read basic science data being presented. You have these wonderfully complex figures. This is a figure 2 from a paper about the dynamics of androgen receptor and estrogen receptor activity in breast cancer. And you have a concept you want to present here in your graph where it's that AR inhibitors diminish estrogen receptor genomic binding. You have different ways of looking at this and quantitating it and displaying that this is true to what you're trying to show, the biological principle of this. And I'm going to focus on figure D here for a minute. Let's get, there it is. Okay, so here is figure D. And what they did was they were using MCF7 cell lines, and they were adding estrogen and then adding certain drugs that were inhibiting the androgen receptor, the enzalutamide, I think that is. Anyway, the point is we look at this and we see, oh my goodness, when you add drugs that block the androgen receptor, that it actually does have a big effect. You get 50% less, as you see in the groups here. You have a much decreased binding events being detected per 1,000 cells. So that seems to be quite a big effect that this does go to prove their effective androgen inhibition on estrogen receptor binding. But let me show you this figure. I've actually doctored it because this is what the original figure looked like. They actually included some results of statistical tests they did on the data. And the reason I presented it the other way beforehand is that I think you, just looking at the data, you could see there was a difference. And I don't know if a statistical test necessarily made it more important or not, or if you see they have their 4 asterisks there to indicate that the decrease in this first group was a p less than 0.0001. Now, I hate to say it, misuse p-values. They misinterpret. I too often hear people will say that we studied drug A and drug B, and drug B is more important because the p-value was so small compared to drug A. And that's not the point. The p-values are not here to tell you that it is important. It's to point the direction to something that might be important. What's important here is the difference that you observe in the data. It's that there was a decrease in the number of binding events here compared to with estrogen alone. It's not this p-value that drives or makes it important. It's really more of a tool like your IncuSite or your microscope that you would use to study the biology. Now, I actually worked with a person who many years ago had to work on some statistics for his results, and he complained to me and said, you know, I never had to do statistics before on my data. We just showed them these graphs. We showed them the various data that we did from our various techniques. And we felt that this is really convincing. And why do we need statistical analyses? And like this nice anonymous statistics professor once said, you don't need statistics to prove a rock can break a window. Now, I'm not trying to talk myself out of a job here. because I think statistics appropriately interpreted and applied can actually be an important tool that you can add to everything else that you're using to study the biology of cancer. And in fact, I just wanted to, I ran a little bit of some data myself where I went into PubMed and I just searched for p-value in papers in the journal Cancer Cell. And I was looking for research papers. And back 20 years ago, only 70, 80% mentioned that they use p-values. And you go up to more recent times, it's going up now to 100%. And also in physiology, they looked at it, and the majority of papers, vast majority, use p-values. So even if you think that you can demonstrate everything nicely with your wonderful data from your results of your different experiments, I think that papers and reviewers are still going to ask you where that p-value is. And so I think you're going to be stuck with them. But as I said, they can be used and can be useful for you in your search for studying the causes of cancer and such. Now, I'm going to divert a little bit and talk about cell lines. And I'm going to preach a bit to the choir here because I know people who do cell lines understand the next two slides I'm going to talk about that cell lines are great advantage. They've been a great boon to research, to trying to understand the biology of cancer and other diseases, that they're cost effective. You can grow what you want according to your resources. And they're really not ethical concerns. You don't have to worry about the PHI attached to your cell line. But this is a paper I quoted here. This is a quote that came from that they said that cell lines are a pure population of cells that provide consistent samples and reproducible results. Now, they set that up as a straw dog because then they tore it apart. They just meant to say that that's what people may think of cell lines. But as people who do cell lines know, that there are actually a number of problems that in using cell lines. This is a paper that was talking about the effect of these immortalized cell lines who may have been around for years or decades are probably have issues that don't make them as useful if you're trying to estimate the biology of the original tumor or the primary cells. And they, in fact, were saying that you should use more tissue more closely related to the patient, like patient-derived xenografts. But what was interesting in here, I didn't actually realize that cells, cancer cells in culture are adapting. They're changing. They have mutations where they are adapting, I think, to their life in this rarefied environment of being in flasks. So given these disadvantages, I'm sure you know about them, and don't really have to tell you about it. But there are other problems, I think, as a statistician, approaching and looking at the data, that when you apply classical statistics to some of the data from cell line studies, that you do have some issues that don't quite gel, don't match what you really would like to do. And in this talk, we're going to talk about the issue of generalizability. We'll explain what that is and why I think that's a problem. This is a more theoretical part of the talk. Then we're going to go on about lack of independence. I'll talk about dependence in cells and how this can lead to biased results like false positive results. And small sample sizes, again, which can lead to a lack of power and to some false findings. And we'll cover each of these in turn. So as an example, kind of maybe a whimsical example, say that we have a rare cancer in Colorado called yellow spot cancer, maybe related to Rocky Mountain fever or something that people develop. And you have an eager young researcher who is eager to understand this cancer. And she has actually found every person who was diagnosed with yellow spot cancer in 2021 in Colorado, as represented by all the dots in this little circle. Now, she wants to study, and since she knows what the population is and she can study everybody, she could just go and collect information from every person and use that and do her studies on the material information she collects from the entire population. But that's not really practical, and that's not how we do research. We work with samples. We really don't want to run all over the state and go to Grand Junction and everywhere to collect samples, where we will deal with samples that are going to be easier for us to, given limitations of money and time, that we will deal with samples. Say we deal with people from the University of Colorado Hospital, and we study the yellow spot cancer population there. Now, when we do samples, the main idea when we do them is we want to use the sample as a surrogate for the entire population. We want to be able to infer from our findings from the sample back to the population, because we don't just want to help the people from our sample. Our goal is to understand what's happening in the entire population. Now, the way we look at it, we talk about inferential statistics, because when you have a sample, you can determine like the mean age or mean levels of gene expression that you're measuring in the sample. And these are called statistics, and they're usually indicated in formulas using Roman letters like M or X bar. And what they are is estimates of the truth in the population. When you look at the population, and we're talking about population data, like the mean age, for example, or mean gene expression, we use the Greek letters. So for mean, we would use mu. And that is the truth. Now, this researcher could get the truth. There is an actual value of the mean age of every person diagnosed with yellow spot cancer in Colorado in 2021. But often we do not know what the truth is. We only can use our samples to estimate the truth. So in the basics of inferential statistics, the current dogma is to use something called null hypothesis significance testing. Gee, I can make that into a tongue twister, say that 3 times fast. But what you do with that is, say your question in your sample, as an example, is to make inferences from the sample to the population. You actually want to find out, you hypothesize that men are diagnosed younger with yellow spot cancer. And so what you do in your sample is you calculate the mean, and you calculate the mean for men and mean for women. And as shown in this is the t statistic for an independent t-test. And here you've got the Roman letters in here. This is the mean for group 1, which could be men, and this is the mean for women. Here we have the standard deviation, the variability of the data, and we have the sample size for each group. And you would plug your values that you determined from your sample into this, and you calculate a t statistic. And we're going to talk about this a little bit more later. But from this, you then can determine what your p-value. Generally, the higher the p-value, the higher the t statistic, the smaller the p-value. But then what you do, and this is something that Fisher, one of the fathers of modern statistics, set up years and years ago, was that he thought we can use data to help us determine what might be important in our data, in our research. And what he did is he set up a kind of a straw hypothesis called the null hypothesis that you would knock down or not. And what it is, the null hypothesis saying there's no difference. So we're saying that the mean age of males is equal to the mean age of females. And then you have an alternative hypothesis, which that ages are not equal. So what you do is you take your data, your statistics you calculated from your sample, you plug them in here, and adjusting for your sample size, your degrees of freedom, you then come up with a P is either going to be less than 0.05, which is your level of significance, it's commonly set at 0.05. You reject the null hypothesis and accept the alternative. So you then conclude there is a difference. If P is greater than 0.05, you fail to reject the null hypothesis and end up concluding that there is no difference in your sample. But I want to point out in this slide is that the null hypothesis here in the formula is not driven by our sample. What we are doing here is we are making our inferences about the value in the population, not about the value in our sample. So what we are trying to do in this is from our samples is to make inferences to the population that we want to study. Also, in common assumptions that when we do for inferential statistics, that we do have assumptions about the tests we're going to use that do differ depending on the test. I'm going to stick a little bit with the t-test. But one of the things is that you do have random samples, because if you have non-biased sampling, then you're going to increase your probability of having a sample that reflects what the general with the population you're studying. So you have more generalizable results from your sample to the population. Independence is also an important data that your samples or your data are independent. Normality and normal distribution or equal variance, which we can also call homoscedasticity, which is a wonderful word. I use it often. But I've highlighted here the 2 concepts I'm going to talk about in terms of generalizability and then independence with these balloon dogs as we go on. We will touch on normality and equal variance later. Okay, generalizability. I just Googled general and I came up here with a nice picture from Wikipedia of George Patton, a famous general from World War II. But then I also want to keep here a reminder of what our generalizability is and its relationship to the population we want to study. So we're asking, does our sample represent the entire population? There's almost always sampling bias when you do a sample. It's not going to exactly reflect. You can do repeated samples, even random samples from the population. You're going to get slightly different, or depending on even big differences. If you repeat an experiment a number of times, there is going to be bias, but you really want to try to represent the biological, and if it's socioeconomic, whatever diversity of the population of people who have yellow spot cancer. And so you really want to make sure you're capturing biological variability or other variability that you are studying, or maybe your results are going to be biased in this. So a random sample where people in the population have equal chance of being chosen is probably the best way, but that is often not possible. We don't know what the whole population is, so we can't really do a true random sampling of that. But often what we end up doing is a convenience sample. And it's something that is there, like we have that lovely University of Colorado hospital right there with samples that we can use and data that we can mine. And it's wonderfully convenient. You can just walk down the campus and you're there. You can meet with the physicians and nurses and people that could help you with the data, who can consent patients and such. But the question is, this is a lovely convenient sample, but really are all patients at University of Colorado Hospital representative of the population of patients in, say, Colorado or Denver or the rest of the United States? Take an example of prostate cancer. Do the patients who come to University of Colorado for prostate cancer treatment, do they represent all patients who are treated for prostate cancer in Colorado? I have some indication that they aren't, because I've been developing a database where I'm matching our archive of prostatectomy specimens to certain clinical data and parameters, including if they have a biopsy that was performed at University of Colorado Hospital. And I'm finding that less than 40% of people who have prostatectomies at University of Colorado Hospital actually have their biopsies done there as well. Because the usual standard of care with prostate cancer is you start with some screening, like a positive PSA test or a abnormal digital rectal exam. And then from that, you may go on to biopsy. And if they find cancer, then you can go on to either watchful waiting or treatment modalities, radiation or prostatectomy to treat it. So a lot of the care, the patients are being referred to University of Colorado Hospital. They're coming from other areas where they're being treated for their early stages, and people who then don't need or watchful waiting are probably still having their care elsewhere other than University of Colorado Hospital. So we are probably seeing patients who require specific types of treatments, who may be more sick, who may, because we are a reference hospital, we draw from the region. So we may not represent for prostate cancer, the biological or the socioeconomic variability of prostate cancer. Does that mean we have a bad sample? No, it just means maybe we need to acknowledge that when we present our data in papers, that we may not represent the full scope of prostate cancer. Now, how about with cell lines, sampling in cell lines? Let's talk about deriving a cell line here and where sampling bias or error may occur. So you're going to start a cell line. You start with, we'll use breast cancer here as an example. You're going to choose a woman with breast cancer from the population of all people with breast cancer. That's a sample of one. In order to get some sort of biological diversity here, you're going to have to sample a bunch of women and create a bunch of cell lines. But in the case we're making one, okay, then you want to use the tumor to create your cell line, but you're not going to get all the tumor. You're going to, it's necessary and critically important that we derive information from the tumor through doing pathology on it and histochemistry, looking at the architecture to determine grade and severity. Also to get idea about receptor status to help determine the appropriate treatment. So you're getting part of that tumor. So you're getting some sort of subsample of that tumor. Then from that, you're going to derive your immortalized cell lines by getting your primary cells and then modifying them by chemicals or viruses so they become immortal. Now you're probably selecting for more proliferative cells. So you might be getting a bias to poorly differentiated cells. But then once you establish the cell line, there is a continuation of perhaps some strain development based on number of passages, a number of or conditions for your media or how you are growing the cells, and they're changing. So when you get down to here, for example, you have a very different population of cells than you see from the original cell line, or you would probably even see from the original tumor. So you're getting this constant change in evolution or selection of cells. So it's wondering when you get to this point is how well these represent the original cell line or how it represents original tumor, or even the original population of breast cancer people. And there's heterogeneity everywhere in here where you have many different types of breast cancers. Does your tumor, is it the most common, your invasive ductal, your IDCs, or are you looking that's an invasive lobular? Also, you have intratumoral heterogeneity where you have different cell populations within the tumor. And when you get your sample from where they gross it out, what are you getting? Are you getting a full representation of the tumors within that, of the cells within that particular tumor? And then once you manipulate the cells to be immortalized, say you come up here, you end up with the MCF7, which is an IDC tumor, a metastatic site derived from a pleural effusion from a 69-year-old woman. So again, you ask the questions, how well does MCF7 represent all breast cancer patients? How well does it represent the original tumor the patient had? And as you go through the maintenance and different passages and culture conditions, how well does MCF7 actually represent MCF7? And I know this is a big issue now, and people will have to, in their papers, make sure that they authenticate their cell lines and also determine, like if you have an ER positive cell line, that you want to make sure that what you have there is still ER positive, if that's what you want to study. So you guys are aware of this, that you know that that's important. And to bring this down to a little bit more personal level, here we have Henrietta Lacks, who was originator of one of the most abundant and used cell lines, used for many things, not just cancer studies, but developing vaccines and such, that her cancer cell lines have been around for now. She died in 1951, so it's been 62 years. So we've had these things in cultures for decades. And how well do her cells, I mean, she's made an immense contribution to science, but how well do they really represent, say, all cervical cancer patients? I mean, she died at 31, her average age is 59. And then how do these cells, how have they changed over the years? And how can we generalize from what we're studying in these cells back to Henrietta Lacks or other women with ovarian cancer? And just as an aside, I have no financial interest in this. I got this nice scanning electron micrograph picture of HeLa cells from this art site. And I thought it was rather funny that you can actually buy a phone case, if you like, of the HeLa cell. Feel free to explore that if you want. Just a interesting aside. So again, this is now going back this theoretical, what is the reference population? And when we're doing inferential tests, we're making references back to the population. So say we start with these cells have gone through many passages and culture conditions, and we have a different strain of that original cancer cell line. And then we do, as traditionally, we do duplicates. We have duplicate cells in a multi-well plate, and we do multiple experiments, and you get results from this. But what is the reference population we're talking about? Is this referencing back just to this population of these cells? I mean, are they different enough from the original cell line or from the original tumor or from whatever cancer it's studying at the beginning? Are they different? I mean, what are we studying? And I do ask people, what is the reference population? What are you trying to make your conclusions about? And this is something that kind of has bothered me for some time as a statistician. Also, when we're talking about in animal studies where you're doing cell line xenografts, where you're taking a cell line and you've done a suspension and you're able to inject a kind of known quantity of cells into the flank of animal or both sides, and then you randomize the animal to either treatment or not, and you watch how this tumor grows. Now, when we talk about issues of experimental units, which is a different talk, but related to this, that when you want to determine which is the animal or the unit that is the unit of interest, the experimental unit usually represents the unit that has been randomized to treatment and is often what you are wanting to make your conclusions about in the biology of the cancer. So the mice are randomized to treatment here, but do you really care about what's happening in the mouse? Is that the population you're studying, or is it the tumor? I mean, you can't really separate the tumor from the screen on the back from the mouse. So is it the cell line that is randomized? I mean, it gets a little fuzzy here when I'm trying to figure about the representative population. So we fall back here. If it's the cell line, then it goes back to, well, what is the cell line? What strain is it? How does it really apply to the population? Because I think what really people were trying to, when we do this, is we want to make inferences to the population of people with the cancer of interest. We don't want to make it back to the cell lines that we're studying. The idea is to make it back to the people, but I wonder sometimes if there is a disconnect in that. So this is a conundrum of the statistician looking in the data, is that what really is the reference population? What is it that I am applying to when I'm making conclusions from my inferential statistics, when I'm interpreting those p-values? Now, independence, or probably more, I should be more likely dependence, because it's really, this is more of the issue that can be problematic. Data or subjects are independent when, like, say you do one measurement in each subject or specimen, and each subject or specimen are not related and they don't influence each other on the level of the measurement that you're taking from them. Dependence occurs when you do multiple, let's say the same measurement on the same person multiple times, or the same specimen multiple times, either at once, which you might consider technical replicates, or over time, if you want to look at changes in values over time. They are going to influence each other, so they're not independent, they are dependent. Now, why is this important? Well, what you happen is that if you have values that are dependent or correlated, you're going to have less variability. And I'll go into a little bit more detail where that's a problem. But for example, you want to do a genetic study. Are you going to, if you study unrelated people, I think you can assume you have independence between that. You hope that one person is not going to influence the results. If you're looking at genetics between brother and sister, you have issues of they do share some genetic material, and also they share a similar environment that could influence your results, where the results that you would measure within these people would be somehow related. And if you use monozygotic twins, unless it's specifically about monozygotic twins, you probably don't want, these are going to be extremely similar type of results. Or in a study here where you were looking at changes in cholesterol level after you've given a drug over an 8-week period, sorry. And what these lines represent is each individual patient in the study. So what happens, it looks like the drug does make an effect, and everybody seems, these lines are fairly parallel to each other. But if you're high, you start out with high cholesterol and you do get a decrease, but you still have a higher cholesterol than, say, this person who started out with a lower level of cholesterol. They still went down, but it stays low. So what happens is the variability that you're measuring with these repeated measures is less than the variability if you treated them as independent observations between the people. And why this is a problem is, let's look at this T statistic, where you calculate your mean of each group. So you've got your standard deviation. Again, let's use the age of diagnosis of yellow spot cancer. And what you would do is you calculate your statistics, and the way that your T statistic increases, because the greater the T statistic is, the smaller the p-value, and more likely you are to cross that level of significance, is you can increase the sample size. Because what this does is that this makes this term smaller, and this whole term in the denominator is smaller for the calculation of T. That will make T larger. You can increase the difference in effect size. If you're finding a bigger difference, this term is going to be larger, and so T will be larger. But also variability in your data. If you have little variability, that's going to make this term smaller. The whole term will be smaller, and thus the T will be bigger, and you're more likely to get a smaller p-value and get that, cross that level of significance. So knowing whether you have independence or dependence in your data is important, because it will influence your study design and also help determine your sample size, but also the type of test that you will use. Dependence is a problem because your dependent observations have reduced variability and they can introduce bias, where if you have reduced everything else remaining the same and you just have introduced dependency in the data that you didn't realize, you're going to have less variability that will make your T bigger, and you are then subject to a type 1 error or false positive result. So here, looking at some in vitro laboratory studies, it's typical that you do 3 replicates when you are plating your values. I guess now it's more likely you're going to do random, not in these nice neat rows, so you can try to reduce plating effect. But for this purpose, we're going to just that you plated your cells. So you created your cells, you made a suspension where you know the number of cells that you're putting into each well of the plate, and you have a level of a drug that you're using to determine the viability or cell growth at different concentrations of the drug. So you have one cell line, 6 different drug concentrations done in triplicate. Now these triplicates are related. The ideas are not meant to be independent observations, that these are technical replicates you're using to determine, are you pipetting the correct number of cells or the amount of treatment solution in there. And it's more to test that your experimental procedure within this procedure is correct. And you expect these or hope that these will be the same and that your technique is good. And then you would just average these, and they are dependent data and treated as such. But the way that we do find a lot of this has always bothered me, I'm going to tell you. When you do in vitro experiments, in order to increase your sample size, you do the same experiment, the same material over different independent experimental runs, say once a week. The idea is that repeating is to show that the effect is robust enough to survive multiple repeats of the experimental protocol, because there could be variations, temperature, reagents, and such. But so this is considered in analyses that this is an N = 3 for independent experiments, and it's kind of the minimal standard for defining N in cell line experiments. But are they really independent? Where is the biological diversity in this? Or how about if you do 3 cell lines? You plate identically, the same triplicates and same experimental conditions in the plate. You would call that an N of 3 because you're capturing some biological variability. But then you repeat the same experiments again over 3 weeks. I guess you would consider that to be an N of 9. I wonder, well, let me go on. I have some problems with that, because what does N of 3 mean? I know it's getting common now to do single cell experiments. And I think that if you're looking at cells from the same cell line or same person, there's going to be a dependency in them. And I have a couple slides to discuss that a little bit. You're doing the 3 culture wells, where you are cell culture, where you're pipetting them into wells in your plate. Technical replicates are dependent. They would be an N of 1. Your 3 independent experiments, we're repeating the experiment independently over 3 time periods. I kind of consider those to be technical replicates. I don't know if I'm being, making people mad with this, but aren't they? You're just testing. You're not really testing biological variability. You're testing variability in the experimental technique. And aren't they dependent because it's the same technique and the same material? It might not this be then an N of 1. I know they're treated as an N of 3, but I, as a statistician, look at these and have some problems with that. And if you have an N of 3 and run your experiments on 3 different cell lines, these would be true biological replicates because they represent tumors from different people. And they would be independent and would have an N of 3. Now, in animal studies, sample size is often the number of animals used, and 5 is a common way to look at them. But there is dependency that does occur even with animals. It might be related to the cage that they're in, or if you're doing animal husbandry, they often talk about lots or pastures or pens of animals. But for example, there is dependency where if you say you want to study neurons and you measure 30 neurons, but from 3 different mice. Now, the problem is the neurons you measure in each individual mice are going to form like a cluster where they're going to be dependent based on the mouse own environmental, genetic, the number of things that are going to make these neurons similar, more similar to each other than to the neurons in another mouse. So the idea is that you have an N of 3 based on the mouse and not an N of 30. This would artificially inflate your numbers. And as remember the t-test, with more numbers, you're more likely to have a larger t statistic and a lower p-value. Now, I've talked to people who do neurobiology and neuroscience, and they say they often consider neurons collected from an individual brain to be independent, and they have ways that they can justify it. And they would then treat the number sample size as the number of neurons they measure. They felt it wasn't ethical just to do one neuron per one mouse, that you use too many mice that way. And so there are issues that do creep into this. And my result, my answer to them usually is, okay, if you feel that is, and you feel you have a justifiable reason to say that the neurons within one brain are actually independent and aren't actually correlated in ways, then justify that. Or alternatively, you do have other experimental techniques that you have cluster analyses or mixed models that you can use, that you can account for maybe the variability within these clusters and do more sophisticated statistical results. But as a rule, you've got a cluster of related data, and that's not going to determine your sample size, but independence will be your 3. Same thing if you're dealing with litters of mice. Say you give the mother mouse a treatment and you want to see how that affected birth weight. So you randomize the mother mouse, she becomes that experimental unit, but that you randomized to treatment, and they have a total of 25 mice. The mice are not the unit you're going to use for your sample size. There are observational units that you're going to measure, and then you will do some sort of mean of the number of mice in that particular litter. So you have a sample size of 4 and not 25. So it's a similar kind of a condition or situation to the neurons. Now, how about when you take a mouse and you put cell line xenografts onto them? Here they're using different cell lines, and what they do is they are different. They are different from each other, and they are looking at the growth of the tumor over time. And what they do is that when the tumors are palpable, tumor growth is determined periodically and tumor volume is calculated. So I think by that, although it was really hard for me to determine because people don't explain things often in enough a way that I can totally understand, it looks like they measured the tumors over time in the same mouse as they represented here. So it was measured about week 1, week 2, 3, and 4. And they said they used 5 mice. So I think, and they show the tumors separately here, which were analyzed by an independent t-test. They treated the tumors as independent on the mouse. And I think this means they had a sample size that for their calculations, they were using 10 tumors rather than 5 mice. So they're treating these as independent. And I've had discussions with people about this often. And there are times where people felt very strongly they were independent. I usually argue they're dependent. They're growing on the same animal. What if this animal is a bullied animal in the group of animals in the cage and is kept from food? If this animal is stressed and isn't getting enough nourishment, that's going to affect the tumors. And it's not going to affect one tumor over the other, but both tumors. These tumors are dependent on the mouse as the host for them to grow. So I don't see how they could be treated as independent. At best, you should do a paired analysis and do maybe the difference in tumor volume as the measurement for this one mouse. So this is another area where you have to be aware. You don't want to artificially inflate your sample size and treat them as independent. So you have 10 independent tumors here, as they did in their analysis, where you should really consider it as an N of 5. You also have technical replicates in animal studies where maybe you take a tumor and you measure it. You can measure the same piece of tumor 3 times and you're expecting to get the same results from that. So you would average your results over that. Biological, if you're doing it where you have independent biological specimens, you take your tumor sample and you're going to measure, and you can do these in triplicates. Say you have 9 tubes overall, it's still an N of 3 based on the number of mice that you've done. And to claim that you have an N of 3 in this is going to lead to pseudo replication, which is going to artificially inflate your N and also will decrease your variability, because these are, you expect these to be similar measurements. So what is that going to do to your statistics? So you're decreasing your S and increasing N. This whole term becomes smaller, and you are then prone to potentially getting some false positive results. This is also in single cell studies. You have a problem of independence and dependence that is not always acknowledged. In this recent paper from last year, they were saying that people will use cells that are studied from the same individual and treating them as independent. But they argue that cells that come from the same individual do share common characteristics derived from their environmental and genetic influences. So there is a greater similarity in gene expression among cells from the same sample or from the same individual. And what they've done is they plotted a graph. They used public data and went through and looked at the intra-individual, so what the variability within a person, the cells measured from a person compared to cells that are measured between individuals. And for each pair of analyses by each pancreatic cell type, they find that the correlation is always higher in the aqua, which represents the intra. So for the individual, you're finding more correlated results. So you have a bias here of finding potentially a positive result if you're just using the cells from an individual and treating them as independent and not accounting for the fact there is dependency in the data. And there are techniques that you can use. I don't do single cell studies, but you can certainly find these out. So the conundrum about dependence is, when I look at the data, are independent experiments performed on the same biological sample at different times really independent? And are bilateral, say, cell line xenografts independent put on the same mouse? And I have people will argue, and I know the dogma is to treat this the same way, but I've always had an issue about that, okay? Now on to very small sample sizes. This is an issue that hit me first when I started looking at data. Is like, you want me to do a data analysis on an N of 3? You know, coming from an epidemiology background, you have you're dealing with populations of people in more larger groups. But yes, I want you to do it on 3. So we've got our 3 independent experiments here. And in animals, it's kind of anecdotally, you know, N of 5 or N of 6 per group is what you do. And my first experience with this was when I was trying to do more of a statistically based analysis of sample size for a group wanting to study kidney disease in pigs. And that's why I chose this cute pig bottom picture to represent that. And I was asking them, what difference do you want to detect? How variable are your data? And that, when the researcher came in and looked at it and said, what are you doing? We always use 5 pigs per group. We always use 5, and that's all we do. And that's kind of the dogma. That's what you do in animal studies. So you are dealing with small groups, and ethically that's good as well, because we don't want to sacrifice animals unnecessarily. But big data, and I'm not a big data person, but I find it interesting that it can also be victim of small sample sizes, where what the biological replicates, that is the individual that you're using, you're maybe doing it in one individual or a couple. And these 2 papers that I found said that you should look at maybe a minimum of 5 biological replicates, or 6, and even more if you want to prove some of your results in some of the big data studies. Now, what happens with small sample sizes is they really suffer from a lack of power. So what happens, you can only get significant results from a small sample size is when you have a very large effect. When you have a small N, you're probably going to have to have a bigger difference you're going to study. A small differences you won't be able to detect. Of course, it's the size of the difference really should be related to if it's clinically or biologically important, not to just how big it is. But with small experiments, you're probably, if you are getting some significant results from your statistics, you're probably finding just large effects. So what power is, it's the probability that your study or statistical test will find a p less than 0.05, if that's your level of significance, if the effect actually exists. So you conclude there is a difference in the groups if it is actually true in the population. And power is, it's often expressed you as in a percentage, you hear it as like 80% power. And it's a function of three factors, including sample size. The easiest way to increase power is to increase sample size. And just because you get a smaller p value because the sample size is bigger, doesn't mean the effect size is any more important. Okay, we have to look at the effect size, how big it is or how small, and also your significance level, which is usually set at 0.05. And here is a lovely power curve where they did a theoretical study in this paper. Actually, it's from dummies.com. I thought it was a great example. And I highlighted the 80% power here because that is usually the level that you want to attain when you're planning for studies. And what they did is they looked at a small effect, kind of a moderate effect, and a larger effect when they're looking at an independent t-test. And in order to reach, with this effect size, in order to reach 80% power, you're going to need about 7 or 8 animals per group. If you have a small effect size to reach 80% power, you're going to need about 28 per group. And sometimes when I worked with people in animal studies and they give me, and I ask them what their detectable difference they want to find and some data variability, and I run these samples and I'll say, looks like you're going to need about 30 animals per group. And they balk because that is extremely expensive and it's also a lot of animals to use. So you really have to look for a larger effect size, because if you look down here at 5, at best in this study, you're going to get 55% power. You're not going to reach this. You're going to have to look for a much bigger effect size when you have small sample sizes. So low power really, having these underpowered studies you're doing and getting potentially false negative findings is one thing that can contribute to your reproducibility in research. So some common data assumptions for inferential statistics, like when you're going to run your t-test, is that you should always check your assumptions, and independence is one thing. You know that from your study design. Is there a normal data distribution? Are your data, is there kind of a symmetrical where you have most of the data in the middle and it tails out? Do you have equal variance in the samples? And the way you test these is you graph the data. You can look at if the do frequency graph is the distribution. You can get distribution of the data. Do the variances appear the same? You do have some formal statistical tests that you can do that would test, like the Shapiro-Wilks test to look for normality, but they're of limited use. In small sample sizes, they don't have the power to detect a normal distribution. In large sample sizes, they're too sensitive. But the ways that we do to try to eyeball and look at these, you can do if you have a large enough sample size. But you can say a small sample size, which is usually considered like less than 20, is not really large enough to test the various statistical assumptions. It can lack power to detect that, or your graphs aren't really going to tell you a lot. So what happens is you don't know. I can't really test the assumptions with small data. So how do I know that I'm using the correct statistical test and not introducing bias by using the improper test? And can I trust a negative result? Or am I making a type 1 error or a false negative error? So these are issues that bother me as a statistician. And to sum up what we've learned today is that meaningful generalizability is difficult, or can be difficult, in cancer cell lines. There's sampling bias, and also the biological variability is not always being captured. There's dependency in data that when you're doing replicates or in how you collect your data or measurement, which can lead to type 1 errors, that's false positives. Small sample size, you can get false negatives and make conclusions that you might miss important results. And also statistical tests may not always be appropriate on smaller sample sizes. It may be difficult to apply. So solutions: do studies with more biological replicates, increase your N. Okay, that's easy, isn't it? Do studies using specimens more closely related to primary tumors. Okay, that's easy, isn't it? Write a novel. Write a lot about your experimental design and statistical analyses. So if you can't do a large N, or you are maybe have some dependency in the data, explain what you did. Use a supplemental information. I mean, like Nature is allowing people to write method sections that don't go into the word count for your main part of your papers, so that you can try to give a more accurate picture of what you're doing. But what my suggestions for you is explain what you did. Explain if you're going to treat both tumors on one animal as independent, and justify it and be detailed. Address potential limitations in your data. Epidemiologists tend to write a whole paragraph about limitations in the paper, but it's important to point out where you've considered that, this may not be generalizable to the population because, and that, and also be specific. I've said this before about each N, because this really helps you for each part of your experiment you're doing. It really does help people coming in and getting an idea of what you're doing, and to judge if what you're doing is correct or if they think it is valid. Thank you for your attention. Are there any questions? I know I threw a lot at you. I can't check the chat, so I don't know if there's anything there.
Kathleen, this is Christopher. Can you
Hi,
hear
Christopher. Yes.
me? On the MCF7, I've been involved in cell line authentication using STR genotyping for years.
I know you have.
And
You've written good papers about that.
over the years, I've worked with a lot of MCF7 samples. There are two papers that have come out in the last 3-4 years. One is by Klingsang at Johns Hopkins and Brown, and the other is by Ben David, and I believe they're at in Boston at Harvard, I think. And in both papers, they worked with MCF7. In the Klingsang paper, they found, using what had been the standard for authentication, the identity of STR profiles at 8 loci. And they found two cell line, they compared two aliquots of the same batch of cell line, MCF7, that had been separated by 3 passages. And they found identical STR profiles, but their responses to drugs were enormously different. The Ben David paper found the same problem and elaborated in more detail. They even went to single cells and found different aliquots derived from single cells of MCF7 that behave differently in response to drugs.
Yeah.
And I have seen in like 20 or 30 samples of MCF7 different profiles. They're related, they're all derived from the same original person, but the cell lines evolve in culture. And I think a lot of people don't realize that. And it's really worth reading those two papers to appreciate that cell lines are constantly evolving. And one misconception people have is if you have an 80% match of STR profiles, they're the same cell line. And that's not what it's meant. It's meant that they came from the same patient, but have evolved doesn't exclude that they haven't evolved.
Yeah, I actually did read those papers. I didn't include them in the talk.
Yeah,
but
it is, MCF7 is just an easy one, and it's one that is a fairly stable cell line, quote unquote.
And is widely used, yeah.
And there are variants out there that people have found are more pathogenic, and they're all treated as equal. And that's, with cell lines, that's a really hard part.
Yeah.
Or hard to accept, because they're constantly changing.
Yep. I had a request in chat. Could you type those references into chat for people to look up?
Mine?
Yeah, the two papers you mentioned.
I'll try to get them.
If not, maybe you can send me.
I'll send them to you.
Or Sir Andrew, and we can disseminate that.
I will send those and a couple others on the problem that I've written about.
Okay. So it makes it even harder to generalize too. What's
Yes.
going on? Because I don't know if the evolution that's going on in the cells and culture is, I don't think it's reflective of what's happening in the evolution of cells in people.
No, because it's different environments.
Yep.
3D cultures seemingly provide a better model if you're going to model diseases, but it is the assumption has been over the years that their cell lines are a stable, unchanging thing, and they're not.
Yeah. That has implications for the biology and also for statistics. Thank you, Christopher.
Yeah, no problem.
I have a comment on the multiple tumors on one mouse. And so it depends really on what question you're asking, because it could be actually an advantage to have one mouse, because if you're comparing the different tumor cell lines or whatever that that on one mouse, they have more similar conditions than if they were in different mice, presumably.
Yeah, each mouse would act, that's like a paired analysis. So if you treated them as a paired analysis and you count for the fact that they are on the same mouse, then yes. Yeah, I think it is valuable that each mouse does act as kind of its own control in that way. I agree. Just don't treat them as independent tumors.
Mm-hmm.
Good observation.
Well, if we don't have any other immediate questions, because you guys reach out to Dr. Torkko. She's usually happy to help there. I just wanted to put a plug. Next month, we have another talk on the 17th with Dr. Zhang to BD on the title, but thank you everybody for attending.
Yes, thank you for listening.
And thank you for giving an excellent talk again, Kathleen. Always listening to you.
Okay, thank you. Oh, flattery, I'll do more. No. Okay, goodbye, everybody.
Thanks, Kathleen.
Okay, bye-bye.